everybody! Today we're going to be going over cell surface receptors. In our last video, we covered the basics of cell receptors, and today we're going to be diving in deeper. So make sure to go check that video out before starting this one. And let's get started. In our last video, we talked about what cell surface receptors are. Let's review quickly. Cell surface receptors are membrane-anchored proteins that bind to ligands on the cell's exterior surface. The important aspect is that the ligand does not need to cross the plasma membrane in this sort of signaling. As a result, ligands can be many different sorts of molecules, including hydrophilic, also known as water-loving, compounds, instead of only being hydrophobic. Each cell surface receptor has three main compounds. It has an external ligand binding domain, called the extracellular domain. Then it has a hydrophobic membrane spanning region, called a transmembrane domain, and it also has an intracellular domain inside the cell. These three domains make up the cell surface receptors. Then there are also three general categories of cell surface receptors. There are ion channel linked receptors, G-protein linked receptors, and enzyme linked receptors. Today, we're only going to be focusing on these last two. So let's first take a look at G-coupled protein linked receptors. G-protein coupled receptors, GPCRs, are a wide class of cell surface receptors with a similar structure and signaling mechanism. Now let's talk about how it functions. When its ligand is not present, a GPCR waits at the plasma membrane in an inactive state. The receptors bind to a special kind of protein called the G protein. What are G proteins? The G protein is a special kind of protein that binds to and hydrolyzes GTP into GDP. G proteins bound to GTP are active, or on, while GDP is inactive, or off. The binding of a ligand activates the GPCR and causes the G protein to exchange GDP for GTP. The now active G protein splits into two, which are then released from the GPCR. Let's take a closer look. Say this is a G-coupled protein. The signaling molecule binds to it, which causes the protein to exchange GDP to GTP. This is the G protein attached to the receptor. Now the G protein splits into one unit and two subunits. It splits into two pieces, and those pieces are released from the GPCR. These subunits interact with other proteins, resulting in the activation of a signaling cascade, which ultimately results in a response. This GTP, in turn, activates enzymes that turn ATP into cAMP, which is needed for the activation of protein kinase A. Let's take a look at the next kind of receptors, called the kinase receptors. Receptor tyrosine kinase are a class of enzyme-linked receptors, which are the third type of receptor found in humans and many other species. What is a kinase? A kinase is an enzyme that transfers phosphate groups to a protein or another target, whereas a receptor tyrosine kinase specifically transfers phosphate groups to the amino acid tyrosine. Now we can go over how this works. Signaling molecules first attach the extracellular domains, which are the ones outside the cell, of two adjacent receptor tyrosine kinase. These two are adjacent to each other, and when they attach to each other, they dimerize. Phosphates are then attached to tyrosines in the intracellular regions of the receptors, and this is called a fully activated receptor tyrosine kinase, a phosphorylated dimer. That's a lot. This is all located in the intracellular region. The signal can then be transmitted to other molecules in the cell via phosphorylated tyrosine. Let's go to an example question. G-protein coupled receptors are a type of receptor that blank. A. Activate tyrosine kinase enzymes. B. Activate G proteins when bound by a ligand. C. Bind to DNA to regulate gene expression. Or D. Are only found in prokaryotic cells. Think about which option you think is correct. The correct option is B. G-protein coupled receptors are a type of receptor that activate G proteins when bound by a ligand. Let's go through the answers and see why B is correct. The first option, A, activate tyrosine kinase enzymes is incorrect because G protein coupled receptors do not have intrinsic tyrosine kinase activity. Instead, they activate downstream signaling cascades through G proteins. Option C, bind to DNA to regulate gene expression, is incorrect because G protein coupled receptors typically do not directly bind to DNA to regulate gene expression. Instead, they activate intracellular signaling pathways that can lead to changes in gene expression. And option D, are only found in prokaryotic cells, is incorrect because G protein coupled receptors are found in both prokaryotic and eukaryotic cells. However, they're more commonly found in eukaryotic cells. And that is it for our video on cell surface receptors. Thank you guys so much for watching. Make sure to like and subscribe.